All right, here we are, we're back. Now we're going to look at osmoregulation and excretion in the vertebrates. Okay, so again, very important to regulate water and salt balance, and they must excrete nitrogenous waste. It's going to get a little bit more difficult to do that. Okay, so recall that nitrogenous waste is just a product of protein and nucleic acid degradation. So as our cells are going through processes and working with proteins, some of the waste product that breaks down has nitrogen in it. And it mainly forms right away, it forms ammonia and ammonium ions. Okay, so it gets more difficult with more complex organisms like vertebrates to get rid of that, because ammonia and ammonium ions are very toxic, but they diffuse with water really easily. Right? And so in some organisms that are real simple cell layers and things, it can diffuse right into the watery medium. But if it's a more complex organism, even if it's living in the water, it can't diffuse. Remember, there's too many thick tissues. So it can't diffuse across all of those tissues into the water. So we need more complex organ systems to do that, as well as vertebrates. We're on land. We can't diffuse it out into the air. It doesn't work that way. You need water as the transport medium, right? So there's a couple ways to deal with that. If the organism can't diffuse the ammonia or ammonium ions immediately, we're going to see that they're going to expend some energy to convert it into one of two forms. It can convert it into urea, which is a much less toxic form than ammonia, and bodies can tolerate it for a little bit longer. You can store some of it in the body. It still requires some water to excrete, though. Okay, so again, we're going to see this kind of cost-benefit analysis, right? Or organisms can convert it to uric acid. Uric acid can be stored in large quantities in the body. It can really be tolerated. It's the least toxic form. Okay, but and it needs very little water to excrete out of the body, right? But it takes a lot of energy in order to make, right? So there's that cost-benefit ratio there analysis, right? So let's look at some examples, and we'll come back to these three forms and kind of look at what I just talked about how much energetically expensive it is, how much water it uses, and how toxic it is. But first, let me give you some examples with marine fishes, or I shouldn't say marine, freshwater and marine fishes. So on the first one, you can see, so these are osmoregulation in fishes. In the first one at the top there, the freshwater fish live in a hypotonic medium, right? They live in fresh water, so it's hypotonic. It has less solute or less salts than their body does, right? So I wrote there, the problem in the fresh water is the body is saltier than the environment. That means that water is just attracted into the body of the fish. Water is gained by osmosis through the tissues. Right? And then it can also lose salt as well. Okay, salts diffuse out. So this is a big imbalance. Just left without any osmoregulatory structures, then this fish would just gain way too much fresh water and lose too much salt and just kind of puff up, and it wouldn't be able to survive, right? So what they do is they do have kidneys that do filter the blood, and they end up producing large amounts of urine in order to get rid of the excess water, right? So they're just taking in fresh water constantly by osmosis, so their kidneys are working to get rid of that excess water. It's only maintaining enough for its cells in order to keep that balance, right? And then it's going to need salts, right? Because it's losing salt by diffusion all the time. So the gills, these are actually really delicate structures. So as it passes water through the gills can actively transport salt into the body. So actively transport means it's using energy to do that. Okay. The gills can also be used to excrete the nitrogenous waste as well. So remember, the gills are part of the circulatory system. Okay, and so any nitrogenous waste can also get excreted through the gills. So the kidney is mainly getting rid of the excess water, again, because of this freshwater environment. The next fish down, he looks the same, but this is indicating a marine fish. Okay, a fish that lives in the ocean. And this one's actually a bony fish, because the one below it, the shark, is also a marine fish, but it's a cartilaginous fish. So they do things a little bit differently. But both of them live in a hypertonic medium, right? So the ocean is much more saltier than their bodies. And so they end up losing water by osmosis. I saw the freshwater fish gained water, right? But remember, so if the body is more hypotonic to the ocean, right? The ocean is hypertonic, means has more salt. Water wants to move toward the salt. So in this case, the middle fish, water is moving out. They're losing water. Okay? And that means that they're gaining salt. They always have salts inside their body. If there's more salt outside in the ocean than inside the body, then salt's going to want to move in. Things move from the area of high concentration to low. So it's high concentration in the ocean, less in the body. So salts move in, waters move out, right? So here what they have to do is, because they're constantly gaining salt, they have to release some of the excess salt. They can't have too much salt. So they do end up excreting it through the gills. Right? They also tend to, their nitrogenous waste, they make ammonia. And since they lose water by osmosis, ammonia can just go with it. So ammonia is easy to get rid of out of the body. It just diffuses out along with water, either through the tissues or through the gills. So they have a kidney, but there's really very little kidney function. Okay? Water and the salts they're able to deal with that just through their tissues and through the gills. So make sure you know the direction, which side of the body is more salty, the ocean environment or the body, and then which way is water going to move. And then how do they deal with salt and ammonia? Okay. The last one in the sharks are cartilaginous fish. So sharks, rays, those types of animals. Okay. So they also live in a marine environment, right? And so the environment is saltier than the body. But what they do is, for their nitrogenous waste, they convert it into urea. So remember, I mentioned urea is a type of nitrogenous waste that can be tolerated in the body at high levels. So they actually retain their urea within their body. Okay, and so they keep it within their body, and that ends up making their body more hypertonic than the seawater. So they reverse the situation than the bony fish do above it. Okay, so again, they retain the urea, keep it in their body, and then that makes them hypertonic to the seawater. So now you can see the arrow direction shifted for water. Water is actually gained through osmosis because their body is saltier by retaining their urea. Okay, and so water moves to the area where there's more solute. Again, it's not always just about salt. It can be other things too, other solutes. So remember, solute is just a general term for some kind of molecular particle. Okay, so water enters the shark by osmosis. And then this extra water, though, it can excrete a large quantity of dilute urine. So there's a high kidney output, and also salt excretion as well. Okay, mainly through a gland. They have an extra gland to do that. Alrighty, so again, make sure you know how freshwater and marine fishes, and distinguish between bony fish and cartilaginous fish. How do they deal with water imbalance, salt imbalance, and then also the nitrogenous waste? Okay. Alrighty. And here's a little a slide that kind of summarizes a lot of things, but then introduces the dry land organisms as well. So remember, nitrogenous waste can be formed as either ammonia, right, urea, or uric acid. Ammonia is the most toxic. That's what you see below each of the panels. Ammonia is the most toxic. Urea is in the middle, has less toxicity than ammonia, but we can tolerate building some up in our tissues. Uric acid is the least toxic and can be stored for a while in the tissues. Okay. So because ammonia is the most toxic, it gets excreted as soon as it's formed, but you need a lot of water to do that. So therefore, we just saw that example. It's most of the aquatic animals do that, especially the invertebrates and things in the water. So as soon as it's formed, it just diffuses out of their tissues. Okay, and because of that, it's just simple diffusion. There's no energy required. So the cost is the toxicity, but the benefit is that there's no energy required. But again, so they have to be in the right environment to do that. Those on land, so mammals, most amphibians, remember they're partially on land and in the water. Some of the marine fishes and some reptiles and terrestrial invertebrates. So they can't tolerate the toxicity of the ammonia. They're not in a medium, or they're in a different type of a medium like you just saw with the other fish, where they can't, water actually comes into the body in the watery medium, or we have to conserve water in our body. So we can't just diffuse ammonia out of our body. So these organisms convert it to urea, right? So again, urea is less toxic. In order to excrete it out of the body, it doesn't need as much water as ammonia, but it still needs water. Okay? And then it requires ATP in order to convert and make urea and even retain it in the body. So it does require some energy expenditure. So again, it's that cost-benefit analysis. So we're expending some energy, we have a little cost, but in order to retain a molecule that's less toxic than ammonia, so we can tolerate it a little bit more. Okay, and so within the graphic there, you can see the table. The kind of gives you the energy required for production, and ammonia was none, because it just happens with the breakdown products of protein and nucleic acids. But those things we need to convert to urea. That's got moderate amount of energy to produce. Okay, you can see the amount of water required for excretion. So we just want to look at relative differences between these three. Okay, and then some animals like birds, insects, most of the reptiles, they produce that uric acid, right? So that's that white material. If a bird ever pooped on you, for the most part, it's pretty thick. It doesn't have a lot of water in it. If anybody owns snakes or other reptiles, you see that white powdery stuff come out. It dries pretty quick because there's not a lot of water inside of it. Okay, so it balances conserving water, right, and making a really very non-toxic form of a molecule that can be held within the body, but it's really costly to make. Takes a lot of energy to convert the nitrogenous waste into uric acid. But again, look at these animals. So a bird is up in the air flying. It can't always take the time to land and go to the bathroom. They do have to land to go many times. Some birds do have to land in order to release it. And so they can just tolerate it inside of their body for a while, right? And they may not have access to water as quickly as other animals might do. So that helps to balance that water. And what about our reptiles? They live in very dry desert areas, right? So they're conserving a lot of water to do that. So again, it takes a lot of energy to convert, but it might be worth by saving so much water in the process. Okay. All right, so let's look at the mammals. What about the mammals? We've got this urea. How do we make urine? How do we balance salt and water and excrete this nitrogenous waste out of our body? Right, so we have an organ system called the urinary system, or it's called the excretory system. Either one is correct. So we use an organ called a kidney. in order to use as filtration organs. So these kidneys, you can see, I have a picture of one of them there. There's a pair in the picture in the background. We have a pair of kidneys. You only need one to function correctly. So if you lose a kidney, you will be able to continue to filter your blood normally. Okay, but you need at least one. Okay, and we'll talk a little bit more about that later. But you can see there's the blood supply associated with the kidney. So the kidneys receive a large proportion of our blood in order to filter and clean it out, in order to balance the amount of water with salt, and then excrete the waste products that we don't need, or the excess water as needed, right? So you can see the blood vessels branching throughout the kidney, okay? And we're going to look deeper and deeper into the kidney, but for now, realize that there's kind of two portions to it. That outer region is called the renal cortex, and then the inner region, which looks like those pyramids, those triangles in there, that's called the medulla. And that's important because the tissue is slightly different in each area, and that's going to be important for which way water moves. Okay, so within the kidney, we're going to look really closely as to how urine is made. But for now, to go through the rest of the organ system, once urine is made, you can see that there's a tube connected to each kidney called the ureter. So that collects any of the urine that gets made to be excreted out of the body. So that collects it and conducts it down to the urinary bladder. You see that right at the base of the pelvis. So that can store the urine for quite a bit of time, right? Some of us can store it longer than others. It's a muscular organ that can really expand really large. And as more and more urine gets conducted down in there, it stores it until you're ready to go. And the tube connected to the urinary bladder that extends out to the external environment is called the urethra. Right? So in women, it's pretty short, the urethra. In men, the urethra extends through the length of the penis. So it's a little bit longer there. Alrighty. So let's go deep into the kidney, and you can see there's a little box there that's boxed out, a little yellow twisted tubule, if you can see that. It's called the nephron. That's the functional unit of the kidney, is the nephron. So our kidney is just showing one there, but it has like 1.25 million nephrons in each kidney. That's how many there are. And this is what one looks like up close. All right, so we say it's the functional unit of the kidney, because this is what is forming urine. Okay, so again, there's 1.25 million of them in each kidney. Okay, and that's what's necessary in order to filter the blood, and then have what's ever filtered go through these tubules. Remember, it looks really very similar to that metanephridial tubule. Okay, so we're seeing a similarity there. Okay, you notice that the little two red structures going into that glomerular capsule, that's the blood system. Okay, so it's cut off. You're not seeing the blood portion. You're just seeing the tubule portion. So we're going to look closely and see those two. But notice that this tubule is all twisted around, and there are different segments of the tubules actually are created out of different cell types, which are specialized to deal with a different portion of the fluid. It might work in reabsorbing glucose, or amino acids, or sodium, or water. So different areas of the nephron do different reabsorb or secrete different portions of the fluid that's been filtered, right? Because of the specialization in different parts of the tube, right? So that first part is called the glomerular capsule, and we're going to look closely at that in a minute. That's where the filtration takes place, because that's where the blood is coming in. Okay, and then that capsule forms that tube. It's all twisted around, and that's called the proximal convoluted tubule. Proximal convoluted tubule, or PCT. Then that branches down into that long loop. It kind of goes down and then back up. That's called the loop of Henle. Okay? And then it twists around again and forms that other section called the distal convoluted tubule, the DCT. Okay, proximal means close to, distal means further away from, and it's referring to the glomerular capsule because that's the beginning of the nephron. Okay. And then you can see the distal tubule connects to that branched area that looks like branched out looking tubule called the collecting duct. So those branches would be connected to other nephrons. Remember, we have 1.25 million of them. So one collecting duct can collect the fluid from several different nephrons. Okay, and then that's the collecting duct. Let me go back to this kidney. Those collecting ducts will collect all the urine through the medulla and then conduct it down to the ureter. Okay, so each of these individual nephrons, 1.25 million of them in each kidney, they're each filtering the blood, and they're each changing the filtration, the concentration of the filtration within the tubule. And what's ever left over in the collecting duct, that becomes urine. Okay, so here's a more accurate picture of what it looks like in the kidney. So you see the two blood vessels, the red and the blue, kind of running horizontally. Well, above that is the cortex, and below that is the medulla. So you can see a nephron starts in the cortex and extends down in the medulla and back up. Okay? And you can see that branching off of that arterial, or that arterial, it branches into that glomerular capsule, and there's a little ball of capillaries inside of there. That's called the glomerulus. And then the nephron portion itself, the yellow, forms that cap over the top of it. So we're kind of looking through inside. You can see the glomerulus. That cap is called the Bowman's capsule. And together, those structures together, that's just called the renal corpuscle. Then the Bowman's capsule forms the tube, and then we get into the proximal tubule, and then the loop of Henle, and the distal tubule, and then finally to the collecting duct. Okay, but notice that the glomerulus, the blood vessel portion, it branches out of the renal corpuscle and then forms another network of capillaries that just kind of folds all around the proximal tubule, the distal tubule, the loop of Henle. Right, so there's a close association between the tubules and the blood vessels. Okay, and that's really important. Okay, and we're going to keep going. We're going to step all of this together. Okay, so here's a close-up of the renal corpuscle. Okay, so remember that the renal corpuscle itself has the glomerulus inside, and that's just a cluster of capillaries that kind of form a knot, and they're under pressure. So when blood comes into the glomerulus from the afferent arteriole, blood comes in that direction. The glomerulus is under pressure, and so you can see, and they're leaky. It's capillaries that are leaky, and that pressure just pushes the fluid out. Not all of it, but it pushes the water along with any solutes that can get through those barriers, and it gets captured by the Bowman's capsule. And then the rest of the blood continues through the glomerulus and comes out the efferent arteriole, and then continues along those peritubular capillaries we just saw, those capillaries that wrap around the rest of the nephron. Okay? So make sure you understand that structure between the Bowman's capsule and the glomerulus, and together they form the renal corpuscle. So this is a site, like I said, of filtration. Okay, so if we filter something, that means there have to be some barriers for the water to be pushed through, which is going to allow some things to pass through, real small solutes to pass through, but other ones to remain inside of the blood vessels, right? And so there you can see that the Bowman's capsule has 2 layers of cells, right? One layer lays over the top of the capillaries. Okay, you can see the glomerular capillary cut right there, and then those podocytes lay over the top. So those are cells of the Bowman's capsule. And then it forms an outer layer forming the cup of the Bowman's capsule, which captures the solute or captures the filtration and conducts it up the top there out to the proximal tubule. So again, think of this, let me go back to this picture, as a big twisted straw. So fluid is coming in through the afferent arteriole. The straw, the Bowman's capsule, that's the opening of the straw. That's capturing what gets filtered out into it and then conducting it down the rest of the straw that's all twisted around into the PCT and the loop of Henle and the DCT all the way to the collecting duct. Okay. So again, those podocytes are cells that lay over the top of the capillaries. So the capillaries themselves are leaky. So water can push through. You can see the little holes that are called fenestrations. Water gets pushed through. Salts get pushed through. Amino acids get pushed through. Red blood cells don't. They don't fit. Okay, so they stay in the capillaries. Even plasma proteins don't fit through. They might start to push through, but then the podocytes prevent them from being pushed through. So plasma proteins stay in the plasma. That's why they're called plasma proteins. Okay. Here's just another picture of what podocytes look like. So, and then you see all the little finger-like projections, and they form these little spaces in between them, and that's where the filtration takes place. Fluid has to squish through there, so it's going to prevent some things from moving through. Okay. So again, here's a picture of the nephron, so you can see it a little bit better. So now you can see, again, the blood vessels on the left, they branch into the afferent arteriole. You see the arrow moving toward the glomerulus, which is the knot of capillaries inside of that Bowman's capsule. So the blood is under pressure and it pushes some of the fluids from the plasma out, taking some solutes with it. Okay, the rest of the blood stays in that glomerulus, and you can see the other arrow. The blood then is conducted out through the efferent arteriole and then continues along the capillary bed, which forms what are called peritubular capillaries. Peri means surround, and tubular, the tubes. So these are capsules surrounding the tubes. So they're very closely associated with the nephron. So anything that got filtered into the nephron, we might have the ability to pull some of those substances back into the blood. You saw that with some of the other organisms, right? We don't want to lose all the water. We don't want to lose all the salts. So we're going to have the ability to reabsorb some of that back into the blood. Anything we don't reabsorb is going to go out through the collecting duct as urine, which is waste, which means we excrete it to the external environment. Okay, so let's look a little bit more closely at filtration. That's the first step, right? So that blood, the afferent arteriole brings blood into the glomerulus. And so 20% of the plasma can leave the glomerulus and filter into the Bowman's space, into that capsule. And we call that the filtrate. Sometimes it's called the glomerular filtrate, might be called the ultrafiltrate. It's all filtrate. It's what was filtered out. So 20% of the plasma. You see, we didn't filter all of the blood, but a good portion of it. And again, each of these nephrons are doing that. So we end up pulling out like 65% of the solutes that are in our blood. We filter that. Okay. So again, proteins and blood cells remain in the plasma because of those filtration barriers. Right? And we actually have what's called a GFR, the rate of filtrate production. Okay? How quickly we filter has to do with the pressure that it's under. But the kidney is really good at regulating the pressure. And so the pressure changes in your cardiovascular system. You know, when you're exercising, your blood pressure climbs. Well, the kidney can regulate that pressure and keep it keep it normal, keep it at the correct pressure at the glomerulus. So it doesn't change the rate of filtration. We want to keep that pretty steady. All those little capillaries are really delicate too. So when you raise your pressure in the rest of your body, rest of your cardiovascular system, it could actually break that pressure. If that raised in the glomerulus, it could break those capillaries and you'd bleed out at your kidneys. That's not good. Right? So the kidney is just able to regulate that. So we have what's called a constant glomerular filtration rate. We can increase the amount of urine production or decrease it, right? But the kidney does that locally. So again, when you raise your blood pressure from exercising or something, it won't affect the rate at the kidney. The kidney will control that. Okay. So that's filtration. We'll leave it at that. It gets a lot more complex, but we'll leave it at that for now. Okay, so that means now that's what the Bowman's capsule has captured. And so from there, it will conduct it down to the first portion of the tubule called the proximal convoluted tubule, right? The PCT. Okay, and here we reabsorb most of what was filtered. So the amount of water that gets filtered, if we just excreted that through urine, that would be 180 liters a day that you would excrete. That's a lot of water. We would die, right? So we end up actually reabsorbing most of that at the proximal convoluted tubule. So again, the cells that make that tubule are able to reabsorb that water. And it also reabsorbs these other solutes, sodium, potassium, chloride, bicarbonate, as well as glucose and amino acids. These are things that are very needed by the body. Your body needs these things. You can see already sodium, potassium, that runs our nervous system, right? Chloride is very important with forming salts with sodium. We need a certain amount of sodium or a certain amount of salts in our body. Bicarbonate is a buffer in our cardiovascular system. Glucose is an energy molecule. You can't afford to lose that. Right? Amino acids make proteins, and most of our body is made out of protein, so we can't afford to lose that either. So those things get reabsorbed. So again, the filtration is indiscriminate. We can't control this being filtered out. So the first portion of the nephron is able to recapture it and put it back in. Okay, and some of it is done, what we do is we can reabsorb sodium, right? And if sodium, we can expend a little energy and pump sodium across the cell membrane back into the capillary. That makes the capillary saltier, right? Well, if the positive sodium goes, the negative chloride follows. That makes it saltier. And remember, water wants to follow salt. So water then moves out of the PCT into the capillary, right? Make the capillary saltier, right? The peritubular capillary, we're out of the glomerulus now. So these are the peritubular capillaries. Pump the salt out, okay? Chloride follows. Now it's salty on the outside, water's gonna follow. So that's reabsorption. Okay, let's look at glucose. We need special transporters to move glucose across. Okay, and so we have what's called a transport maximum. So we have to have these certain binding areas for glucose and also vitamin C. That's another one that gets reabsorbed. Okay, unless the plasma values are so high, then vitamin C will get excreted out through the urine. Okay, same with glucose, but the reason why is because of what's called this transport maximum. Let me come back to this slide. Let me show you the picture. So what we're going to do is we're going to zoom into the proximal convoluted tubule and just look at a single cell. That's what this is. So the upper portion above the cell, right, that's inside of the tubule, right? The lumen of the kidney tubule, lumen means the space inside of the tube. So that's where the filtrate is running through. And you can see there's sodium in there, there's glucose, right? And then on the other side, outside of the tubule, are the capillaries. That's what you see one down there. That's a peritubular capillary, okay, with interstitial space in between. Okay, so in order to absorb glucose across, we need a transporter. And you see that embedded in the membrane of the cell on the apical side of the proximal convoluted tubule. Apical side means it's on the side that touches the lumen. Well, the filtrate is, and glucose is running through there. Well, notice it also pulls sodium. So sodium's attracted to that transporter. It binds, which also then attracts the glucose. So it's a co-transporter. And as they move through, both of them move through the inside of the cell. And then you can see at the other end, next to the capillary, there's another transporter that only glucose runs through, the little purple ball. Okay, and that pops it out to the capillary, and now it's reabsorbed back into the blood cell, or into the blood capillary, right? Into the blood. Sodium can also, again, I mentioned using energy, that's the ATP, get pumped into the capillary. And when that happens, look what's happening. The capillary has a lot of sodium in it. It's got glucose that makes it hypertonic. Water moves through from the filtrate into the capillary by osmosis. Okay, so by expending a little energy to pump sodium through, we recapture glucose and also water. We reabsorb those things. That's the reabsorption process. So the Tmax, the transport maximum, comes into play because those transporters, you see where it says co-transport, there's a whole bunch of them all over the membranes of those cells, but there's a limited number. So if you have a normal diet where you're getting a healthy amount of glucose, you have enough co-transporters to reabsorb all the glucose. So when you go to the doctor and they want to test your urine and they do a glucose test, they're looking for 0 glucose because you should have enough transporters to reabsorb all that glucose. If there is glucose in your urine, that means your diet is probably too high in glucose. Okay, and so you're exceeding what's called this transport maximum. So if your diet is too high in glucose, right, that means we can't capture a lot of the glucose, and then also we lose a lot of water as well because the glucose stays in that nephron. So water, remember, it's got to be more solute, more salt and sugars like glucose in the capillary. Well, if you're eating too much sugar, then that'll get into your kidney, into the nephron, and the water won't get reabsorbed. It stays with the glucose and you urinate it out. So people with diabetes, sometimes they realize they might be getting diabetes because they have a really high urine output. Okay, because they've reached transport maximum. There's not enough transporters to reabsorb all of that glucose. Okay. And that could be associated with, again, diet. It could be associated with type 1 diabetics. They don't make the hormone insulin, they don't make the hormone insulin, which tells the body to use glucose, right? So if you're not making that hormone, it just stays in the blood, the kidney filters it, it's got too much glucose now because the rest of your body cells aren't using it. So there are not enough transporters to reabsorb it, so it stays in the tubule and you excrete it. And again, that's going to have problems with blood pressure and everything else. Okay, so let me go back to, so again, at the PCT, just to review, at the proximal convoluted tubule is where reabsorption takes place. So we reabsorb sodium, chloride follows, right? Therefore, water follows. When we reabsorb sodium, we have transporters that also bring glucose across. Okay, so we reabsorb all the glucose. I used those as an example. It's pretty much the same process with amino acids. So just know that amino acids get reabsorbed. So again, if in a sample of urine, you should not have amino acids in there. Okay, we also reabsorb potassium and bicarbonate. I'm not going to show you specifically how we do that. Okay, so if you can stick with this slide and how we reabsorb sodium, right, and glucose, you should be good to go, and know that water follows. Okay, because again, you're making the inside of the capillary more hypertonic, so water is going to move out of the PCT into the capillary. Okay, the next segment of the nephron is the loop of Henle. Okay, the loop of Henle. Its job is to continue to reabsorb sodium and therefore water. Okay, and notice at the loop of Henle, right, it's continuous with the PCT. So as the fluid moves out of the PCT, which is located up in the cortex area of the kidney, it moves down into the medulla. And that loop, then that first segment of the loop is called the descending limb because the fluid is moving down into the medulla. Okay, again, still contained within the tubule. Then it takes a what's called a hairpin turn. It turns 180 degrees back up. And so now it's part of the ascending limb of the loop of Henle. So the loop of Henle has a descending limb and an ascending limb, and that just dictates the direction that fluid flows. Okay, and its main job here is water reabsorption. And it does that by setting up a concentration gradient between the tissues surrounding the tubule. Okay. And so on the ascending limb, it has the ability to pump sodium out, expending energy. It pumps sodium out, active transport. Sodium is positive. When sodium flows out, chloride follows. So the outside of the tubule now is hypertonic. Hypertonic. Okay, you can see in the close-up of the ascending limb, the red arrow shows sodium and chloride moving out. But inside the tubule, it shows that curved arrow of water because those cells are not permeable to water on that side, on the ascending limb. So water stays in there. But on the descending limb, it is permeable to water, but not to salt. But because the ascending limb is making the outside more salty, right? Then on the descending limb, water wants to move out. It follows where the salt is. Okay, so it's important to know the ascending limb is permeable to salt, sodium, and chloride. It is not permeable to water. But the descending limb is permeable to water, but not to salt. So the ascending limb sets up the gradient. It pumps salt across, pumps sodium, chloride follows passively. The outside now is salty. So water follows out of the descending limb. And the capillaries are right there, so all of that is reabsorbed into the capillary. So we just said, there's the title. The loop of Henle functions in reabsorption of water, okay, by setting up a salt concentration gradient. Okay, that's what you need to remember. The longer the loop of Henle, the more time, the more water can be reabsorbed. So we actually have two lengths of our nephrons, depending on the loop of Henle. You can see the first one there on the left is called a cortical nephron because most of it is located in the cortex, and it has a relatively short loop of Henle into the medulla. The medulla is where it's salty. Okay? The other one on the right, that's the juxtamedullary nephron. And so you can see most of it is still up in the cortex, but the loop of Henle is all down in the medulla. So again, that's very salty down there. So there's more opportunity for water to be reabsorbed out of the descending limb into the tissues. And again, the peritubular capillaries are there. Always remember the peritubular capillaries. Okay, and so we humans, because we have evolved we make sure we have access to water, right? So we have mostly cortical nephrons, relatively short loops of Henle. We have some juxtamedullary, but for most of our nephrons would be the cortical type. If you were an animal like a gerbil that lived in the desert where it's dry and you don't have the opportunity to find water, and you get most of the water from your food, then you don't want to lose water through urine, right? So desert rodents like gerbils and other things, they don't urinate very much. And that's because most of their nephrons are these juxtamedullary nephrons. And this is what an actual gerbil's kidney looks like. All of those white things in the medulla, all of that white, those are the juxtamedullary nephrons. They're really long and they span through there, that bright white. So it's got it excretes a little bit of urine because, again, water excretion, it's obligatory. We have to do that to get rid of the nitrogenous waste. And a gerbil is a mammal, so it releases urea. So it does have to excrete a little bit in order to get rid of that. But they don't have access to water, so they don't drink. They don't have the ability to replenish water like we do. So they get in a little bit through their food. So since they can't drink and replace it, they've evolved to have these really long loops of Henle so they can reabsorb most of the water that gets filtered. Okay. All right, freshwater fish, by the way, have no loops of Henle at all. They need to excrete as much water as possible. Okay. All right, what's the next frame? Oh, the distal convoluted tubule. Let me advance ahead just to show you the picture, right, of, well, that's not a very good picture. Let me go back. See, just so you know where we're at, we can use this one. So you see the, remember the Bowman's capsule, the renal corpuscle there. You can see those. It's connected to the proximal convoluted tubule. That's where reabsorption takes place of water, sodium, chloride, potassium, glucose, amino acids, urea, or bicarbonate, right? And then we go to the loop of Henle, and that's water reabsorption by setting up a salty gradient in the tissues of the medulla. Now we've curved back up through the ascending limb to the distal convoluted tubule. Distal convoluted tubule. So that's what's next. And we're almost done here. Whoops, I went too far. So this is a close-up in the distal convoluted tubule. So again, you can see the cells of the tubule all the way to the left side. That's inside of the tubule, the lumen. Okay, then you see the cells themselves, and then you see the pinkish is the interstitial fluid, and then a capillary, the peritubular capillary. Remember, that surrounds all of the tubules. Okay, and so the function of the distal convoluted tubule is secretion. Right? So secretion, remember, means that this is stuff that's still in the capillary that did not get filtered out. But we need to regulate some of those items and actually secrete it into the tubule so we can get rid of it. So then we can excrete it. So secretion just means from the blood to another portion of our body. Okay, and in this case, it's the nephron. Excretion, if you excrete something, that means from our body to the external environment. So this stage is secretion from the blood to the tubule, and we will eventually excrete it. Okay, so inside of our plasma, we have a lot of potassium. But remember, for our nerves to function, we need to put potassium from the inside of our nerve cells outside. So we need a concentration gradient. That means the interstitial fluid in the plasma has to stay low in potassium. So whatever didn't get filtered at the Bowman's capsule, we can secrete it out of the peritubular capillary. And so you can see that there right in the middle, you see that K, two of the K, they're moving out of the blood and out of the interstitial fluid through a protein transporter, and we're expending energy to do that. We're pumping it out. But we're going to put sodium back in. We're going to reabsorb sodium, and water's going to follow. So we have another ability to reabsorb some water here. Okay, and if our blood is too acidic, it has too many hydrogens, instead of potassium, we can actually secrete hydrogen out, hydrogen ions. Okay. So see, there's secretion of potassium and hydrogen. Remember that. That's moving into the tubule. We can reabsorb sodium. And then if there's a hormone called aldosterone, if that gets secreted into the blood, it targets the distal tubule and a little bit the collecting duct, but it stimulates the active transport of more sodium, right? And bringing potassium in, secretion. And so what aldosterone does is by doing that, then we can reabsorb even more water. So this is a case where the body, maybe you haven't been drinking water very much and you're a little dehydrated. So you will secrete aldosterone, and that tells the distal convoluted tubule, hey, reabsorb more sodium so that water follows. And we can retain it into our body. We reabsorb it back into the body instead of keeping it in the filtrate, which will eventually be excreted. All right, so those are the functions at the distal convoluted tubule. Then the last portion, that's actually the last portion of the nephron, but remember that each nephron at the DCT connects to the collecting duct, and that's what I have circled there in green, a portion of it circled. So the red is the DCT connected to what's circled in green, the collecting duct. So remember, several nephrons can connect to those branches on the collecting duct. So for the most part, whatever gets then moved from the filtrate, from the DCT into the collecting duct is the urine. Okay, but we have one more chance of reabsorbing water if we need to. If you're really dehydrated, your brain will release a hormone called antidiuretic hormone, ADH. And that targets the collecting duct, means that the collecting duct has receptors for the antidiuretic hormone to bind to. And what it does is it causes the cells of the collecting duct to insert these channels called aquaporins. Aqua means water, right? Pores, aquapores. It makes the collecting duct more permeable to water. So because this is down through the medulla and it's salty from the gradient set up by the loop of Henle, more water gets reabsorbed. So it's almost like this last effort where we're about to excrete whatever water is in the filtrate, that's urine, and the body goes, hey, wait a minute, we're still dehydrated. I don't want to lose that urine or that water through the urine. So ADH tells the collecting duct, become more porous to water. And since it's passing through that salty gradient set up by the loop of Henle, more water gets reabsorbed. So that's variable reabsorption. It only happens if the hormone is present. If it's not present, that means your brain has determined we've got enough water in the blood. What's left in the nephron is excess, so go ahead and excrete it with urine. So without ADH, you might have a higher volume of urine. With ADH, you've got a lower volume of urine. Alrighty, so that's how the kidney functions. That's how we produce urine, right? So we looked at the part of the kidney is called the nephron. It's the nephron and all those different parts, those different functional parts that are manipulating the amount of salts and other things. And then I didn't talk about it enough, but there's urea in there. And so urea is going to stay and just be released through the collecting duct and then out the ureter, right, to the bladder and then through the urethra. So we release it there. Now, so basically a lot of blood is being filtered. Okay, and it's doing that in order to, again, keep our blood, keep the toxins out, and keep that water and salt balance correct inside of your blood. So if you're unable to do that, then you have what's called renal malfunction. So regardless of what causes your kidney not to function, it will all cause what's called uremia. It prevents your kidney from removing toxins out, right? And so that can, if you can't remove toxins out of your blood circulates to every cell in your body, and that's where you can have other organ failures and things. Okay, so the kidney is a very important organ. I mentioned we really regulate the amount of potassium in the blood. If there's too much potassium in the blood, then our cells that produce those action potentials don't function correctly, right? Remember, potassium has to leave the cell in order to repolarize. And if there's too much in the blood, then there's no concentration gradient there. Potassium can't leave. Okay, so that upsets your nerve function and also your heart function as well. So we can go on. I mean, there's so many things that can be harmed when your kidneys aren't working correctly. So again, you can function properly with just one kidney, okay? With only 10% of the nephrons functioning, in fact. You can keep this up. So one kidney, you can afford to lose one kidney. It's better to have two. But if you have something wrong with your kidneys and they want to remove one, you'll be fine. But it's when both kidneys are affected. Okay? You have to filter the blood. And so this is where hemodialysis comes in, or just dialysis. There's a couple of different ways to do it, but this one is showing blood dialysis. And basically, since your kidneys can't receive blood to filter, we take a intravenous needle, we put it in your artery so the blood comes out the artery, and it goes into what's called a dialyzer, a structure that receives your blood, and it has filtration within it. And so there's fluid inside of there causing filtration to occur, removing the toxins, balancing the water and the salt. And then what gets returned back into your vein is purified blood or clean blood, right? And then you can drain out the waste product. So you have to go through this. And in fact, if you haven't voted yet, something like this is on the ballot. I voted today, right? And it has to do with regulating dialysis clinics. So I'm not going to get into the politics. You can read it and determine how you're going to vote for that. But dialysis clinics are very important. This process of dialysis can take 4 to 8 hours. So somebody who has to do dialysis, they have to go sometimes 3 to 5 days a week to get their blood cleaned up. Alrighty, so whether we do it this way or you still create urine a little bit, but after we create urine, again, remember that each kidney is attached to a ureter, and you can see those at the top. So we have two, one for each kidney. If you have one kidney, you only have one. That connects to the urinary bladder along the back side, and you can see those openings, those ureteral openings. That's where it opens into the bladder and starts to fill it up with urine. And so the opening at the bottom is the urethra. But what we do surrounding the urethra, right at the base of the bladder, you have just like similar to the digestive system, you have an internal sphincter and an external sphincter. Okay, and you can see the difference between the female and the male, right? But the internal sphincter is smooth muscle. And so again, as the pressure of the urine starts to build up and pushes on that internal sphincter, there'll be an automatic reflex that causes a smooth muscle to relax. And that's, again, where you go, I have to go to the bathroom. But your external sphincter is skeletal muscle, and that's under voluntary control. So you can clamp that shut until you get to the appropriate place to release the urine through the urethra. Right? So the act of urination is called micturition. Okay? And that's it. That's the urinary system. Hope you enjoyed it. I think this last portion became kind of long. Sorry about that. But I just wanted to keep going through and how the kidney made urine. All right. Thank you, everybody.